All right, well, welcome everyone to the Boulder Peptide Foundation's webinar on IP concerns for peptide. We're really happy to welcome everybody to the webinar today. The Boulder Peptide Foundation, I'll just remind you, is a nonprofit, and we organize an annual live meeting once a year in September. This year it is September 28th to October 1st. We alternate between locations in Boulder, Colorado and in Napa. So this year we're back in Napa at the Silverado Resort, and we hope to see you there. I'll just point out registration is open and poster abstract submission is open. So please go to our website for that. In addition to our annual meeting, we do organize these quarterly free webinars to bring our Boulder Peptide community together on a quarterly basis. And today, very pleased to turn it over to Alaric and to Sally, and they will introduce the speakers and host the webinar. So nice to see everyone.
Thank you, Yvonne. Yeah,
Thank you for joining.
and thank you to all the speakers that are participating. We have a very good panel planned for you today with a combination of lawyers from Cornwall who provide a legal IP 101 for you, and we have Jonathan from Almac that will give you a more technical perspective on how to protect IP in peptides. Yeah, before I'll just do a very quick intro and turn the mic over to Alec, my co-moderator to do and co-organizer for this webinar to do his intro, and then we'll proceed with the introduction of the speakers and their content. Yeah, so hi, I'm Sally Wonley, and I'm a member of the Boulder Peptide Foundation's SAB. It's a real pleasure to be here. Formerly, I was also an IP attorney earlier in my career, and that's part of what brought me here with this webinar. I've also had experience in management consulting as an entrepreneur before becoming an investor in life sciences companies. And my connection with peptides is I once worked as head of global BD for a peptide biotech company, Peplib, which, as you may know, recently did a deal with Novartis. So this is a very special community with me that welcomed me in when I was an outsider, and I've since then never left. Alec, please
Thanks, Sally.
introduce yourself.
Yeah, so hi everyone. So my name's Alaric Demarchely. I work with Almac, CDMO, and I'm a business development manager. I'm a chemist by training, and I've been with the company a while now. And same, I was an outsider before joining this team, but I worked in peptides process development and now BD functions. So thank you very much for having us. I'm also part of the scientific advisory board with Sally and Yvonne to organize the Boulder meeting as well and this specific webinar. So I'll just do a very brief introduction of the speakers and then let them take the floor because you're here to see them, not me. But first, I'll remind you that if you want to ask questions, you can either raise your hand during the talk. There's a function for that, or there will be ample time for Q&As at the end of each talk as well. And there's a Q&A function within the webinar so that you can ask your questions during the talk, and then we can go through them afterwards. So it's my pleasure to introduce Magdalena and Rashad from Crowell. I'll let them do their own proper introductions as well, but very briefly, Magdalena is a patent agent and scientific advisor, and she has a pharmacology background. And Rashad's a partner, and he's a chemical engineer as a background. And later on, we'll also hear from Jonathan Loughrey from Almac, who is Director of Chemical Development, and he's also a chemist by training. So the Crowell team, I'll give you the floor, and please go. Please go on.
Right. Thanks, Alaric. Hi, everyone. I'm Magda Cilella, and my colleague Rashad and I, we're from Crowell and Moring, and we will present on the IP Essential Strategic Protection for Life Sciences Innovation. Like Alaric said, we'll have our presentation will take approximately 40 minutes, and we'll move fast, but we'll have some time left for Q&A at the end of our presentation. Okay, Rashad, next slide, please. So our goals for today are to understand the three core IP pillars, which are patents, trade secrets, and trademarks, and how they work together to protect peptide companies. Then we will examine patent scope and claim strategy for peptide compounds, formulations, and manufacturing processes. And then we will discuss way to identify when to patent versus protect as a trade secret, and how IP strategy must adapt as technologies evolve. And then we will also discuss some of the challenges of patenting personalized medicine and patient-specific treatments. And there will be some business takeaway prompts throughout the session as well. So the first portion of this presentation will provide an overview of patents. So what is a patent? Patent gives you the right to stop others from making, using, or selling your invention for up to 20 years from the date you file a patent application. In exchange, you publicly disclose how your invention works. Think of it as a government-granted monopoly in exchange for teaching the world something new. There are three types of patents: utility, design, and plant patents. Utility patents protect the functional, structural, or technical aspects of a new invention. These patents protect how an invention works, what it's made of, or how it is used. Design patents protect new, original, and ornamental designs for an article of manufacture. They only protect appearance and not the function. And then the plant patents protects new, distinct, and asexually reproduced plant varieties. Plant patents cover the right to exclude others from reproducing, selling, or using the plants or its part. And then, as you see here, there's a illustration of a issued patent. The USPTO issues these physical ribbon copies of patents, and they feature a gold seal and a red ribbon. However, since April 2023, patents are primarily electronic, but you can still get a ceremonial copy with a ribbon by opting in when paying an issue fee for a patent application. There are 5 different categories of what patents can protect, and these categories are shown in this table by claim type. You can have claims to a composition, method of treatment, formulation, manufacturing process, and device. Composition claims will cover the peptide itself, which is any sequences, analogs, conjugates. Method of treatment claims will cover how the peptide is used therapeutically. Then formulation claims will cover how the peptide is delivered, and manufacturing process claim will cover how the peptide is produced. Device claims will cover mechanical subparts of a delivered device, for example, an injector. And then every major innovation in your company should map to at least one of these 5 claim types. To be patentable, an invention must be useful, novel, and non-obvious. And to be useful, an invention must provide a specific, credible, and substantial benefit to the public, rather than merely being theoretical, speculative. And then, importantly, it also must be new, which means that it has not been previously disclosed to public anywhere in the world before the filing date of a patent application. So an invention is not novel if it's already part of prior art, which could be any existing patent, printed publication, articles, public presentations or use, or product on sale. And then non-obviousness, third aspect of this, means that invention is not a simple, predictable, logical improvement that a person, such as another researchers in the same field, would easily deduce from existing public disclosures. And then in the United States, there are certain things that we cannot patent, and they include laws of nature, natural phenomena, abstract ideas, generally you cannot patent mere ideas, formulations, or naturally occurring substances, such as substances found in nature. Bottom line is that patents are reserved for new, non-obvious, and useful inventions. And then the benefits of patent protection include protection of your R&D, which is preventing competitors from entering the market for up to 20 years. Another benefit of patent protection is providing an early lead against workaround designs, also forcing your competitors to use desirable alternatives and forcing competitors to invest in R&D to compete. If you don't patent your idea, someone else might patent it. Rashad, okay. Beyond protection, patents deliver 4 key forms of additional value, and I want to make sure that we cover them as well. Patents attract investment capital. A strong patent portfolio signals to investors that your technology is defensible and proprietary. Patents increase market capital. They are recognized assets, and the market values them as such. They give you marbles to trade. Even if you cannot block a competitor outright, your patents create cross-licensing leverage, which means that you have something to bring to the negotiating table. And they generate licensing revenue. Patents can be actively monetized through royalty arrangement. And for some companies, that can become a significant revenue stream. So the key takeaway here is that patents are not just legal shields. They're strategic business assets, and organizations that treat them as such consistently outperform those that do not. And then in the next section, we will focus on understanding the patent scope. So one of the most important things that I want you to take away from today is that one claim is never enough. Strong patents are built on multiple layers. Broad claims are your first line of defense. Narrower claims should be your backup if the broad ones are successfully challenged. Here's a quick example. You invented a peptide that treats type 2 diabetes. Your broad claim covers any peptide that activates the GLP-1 receptor in a specific way. And then you have a narrow claim that covers the exact amino acid of your molecule, of your peptide. If your broad claim is successfully challenged, the narrow claim may still block a competitor from copying your molecule. And then a patent does not protect your product, but rather it protects what's in your patent claims. Think of patent claim like a fence around a property. A wide fence protects more land, but it's harder to build and easier to challenge. A narrow fence is easier to defend, but it gives your competitors more room to work around you. The strategic question should not be, can we get a patent? It should be, how much territory does our patent actually protect? So I like this visual here, the outer ring, the broad claim. The broad claim scope would be any peptide that brings receptor X and achieves therapeutic effect. So it provides maximum protection, however, it has a higher risk of being invalidated. And we have another layer, which is that middle ring, which is an intermediate claim. For example, an exemplary claim would be a peptide with a specific structure of this structural feature that binds receptor X, and that provides a balance protection. And then there's the inner ring, the narrow claim, which is to the exact peptide sequence. And it's easiest to defend, but it's also easiest for competitors to design around. So a strong patent portfolio has protection at all 3 of these levels. And the next point is critical because the rules around broad claims changed in 2023. Broad claims now require broad data. The case that changed them is Amgen v. Sanofi, and the Supreme Court ruled that if you claim a broad category of peptides based on what they do, so for example, your claim is to any peptide, like any peptide that lowers cholesterol by binding protein X, you must demonstrate with data that your patent covers the full range of what you're claiming. So in other words, you cannot describe just a handful of examples and claim everything that works the same way. So what does this mean practically? It's two things. First, invest in your data early. Before you file, generate experimental data across the full range of compounds you would like to protect. Do not file broad and hope to fill in the gaps later. The court has made clear that this will not hold up. And then the second, you can use continuation applications strategically. And continuation applications are follow-on filings that build on your original applications, and they allow you to pursue additional claims as your science develops, and you generate more data. As you generate more data, you can expand your protection in structured and defensible way. The bottom line, broad claims are still valuable, but now you have to earn them with data. The next section will focus on discussing the timeline for filing patent applications and key things to keep in mind. So the United States operates on a first to file system. What it means is if there's two companies that independently develop the same peptide, the one that files its patent application first wins, regardless of who actually made the discovery first. But filing too early, like before your data is strong enough, or too late, for example, after you publicly disclose your invention, can be as damaging as not filing at all. So this table summarizes the timeline for filing a patent application. First, on the left, in the left column, we have the discovery synthesis phase, and this is where it's very important to document your discovery. If you don't, you are risking of having no record of your priority date. And the second item on the left is before any conference, presentation, or publication, a provisional application must be filed to protect the invention, your invention. Provisional application is a lower cost, faster to prepare filing that locks in your priority date, which is the date from which your patent rights are measured. And a provisional filing does not require full detail of a complete application, but if you don't file a provisional application before public disclosure, you might risk losing patent rights in most international markets. Once a provisional application is filed, or the filing is completed, you have 12 months to file a full application. And you can spend more time on research and experiments, but a full patent application must be filed. If you don't, at the 1-year anniversary, the provisional application expires and your priority date is lost. And then also, important to keep in mind that before any product launch or licensing discussion, it's very important to confirm that all key applications that cover the invention are filed. If you don't, and you go on presenting your invention, companies can file before you. There might be also gaps in coverage. So first to file wins, but filing smart matters more. As I briefly mentioned, publishing or presenting before filing can permanently eliminate international patent rights. In the United States provides a 12-month grace period after your own disclosure. For example, at a conference or a publication in a journal, you can still file a valid U.S. patent application within 12 months of that public disclosure. However, most other countries don't have such grace period. A conference presentation, a published paper, even a detailed conversation at a trade show can immediately and permanently eliminate your patent rights in countries such as Europe, Japan, China, and other major markets. So the practical rule, file before you disclose always. Establish an internal pre-publication review process so that any paper, poster, press release, or conference abstract is reviewed by IP Counsel before it is shared externally. It is also very important to maintain a formal IP filing calendar. Patent deadlines are absolute. Missing a filing deadline by 1 day can mean permanently losing patent rights. Every peptide company, regardless of size, should maintain an IP calendar tracking. As the questions, are you tracking innovations? Are you tracking provisional filing dates and their 12-month conversion deadlines? Are you tracking national phase entry deadlines based on international applications? Are you tracking upcoming publications, conference presentations, any product launches that trigger filing obligations? Business takeaway, ask your team this way. Do we have a pre-publication review process? Does every scientist on our team know to notify the IP Council before presenting or publishing new data? And the next slide, and then my colleague Rashad will take over from here. So Rashad, go ahead.
All right, thank you very much, Magda. And thank you to everyone that is tuning in to this webinar. Magda has explained the first pillar of protection that peptide companies should be paying attention to. And I will now talk about pillar 2, which is trade secrets, and also pillar 3, which are trademarks. But let's start with trade secrets. And I think a good way for us to get an understanding of a trade secret is to first define what a trade secret is. So first and foremost, it is secret. It is information that is not generally known or easily discoverable by others. And once you have that secret, you have to take reasonable measures to preserve this secrecy. And so there are steps that are taken, such as having employees in their employee agreement agree not to disclose information, that few people actually know the secret, or that you have saved the secret in a database that only a few people have passwords. Should this come up in a litigation, these are facts and factors that you would like to show the court that you've taken these measures. If you don't take these steps, it's possible that they might not find that you kept it as a trade secret. And most importantly, it has to have value. So it must have actual value or potential value that arises from the fact that it's generally unknown. One example that we all know, it's not necessarily focused on peptides, but is Coca-Cola. Coca-Cola famously has a trade secret on the actual formula for the Coca-Cola product. That is something that they take reasonable measures to protect. Only a few people know it at one time. And we know that there is value that is derived from it. So taking that example and applying it to peptide companies and what you all are producing, you can see that if there's some value from some of the know-how that you all are generating and expertise that you have, then a trade secret might be an option for protection. And so manufacturing know-how, a patent versus a trade secret, how do you decide which avenue to pursue? And so with regard to your manufacturing process, it could be just as valuable as the peptide itself. If the competitor cannot synthesize or purify your peptide efficiently and they cannot compete effectively, even if they know the sequence. And so even if they have your peptide, or if they generally know the sequence, if they don't know the specific steps that you took as a company to synthesize it or to purify it, then they're not going to be able to compete effectively. And so as you are thinking about protecting your new peptide product, one of the things to think about is, what do we protect as a patent? Do we protect the process that way? But that requires the public disclosure like Magda laid out. Or do we keep it as a trade secret so that it's confidential indefinitely? One key difference between a trade secret and a patent is that your patent protection has a finite time period. As Magda stated, if you file your application and it then issues, you have 20 years from the date of filing protection under your patent. With a trade secret, that trade secret can last indefinitely as long as it remains a secret. But there are some pros and cons to both of those strategies. And so here's an example, or some examples of things that might be worth protecting, either as a trade secret or as a patent. So your synthesis optimization parameters, the specific temperatures, the solvents, the coupling conditions, reaction times, et cetera, that your competitors would not be able to determine even if they have your final product or your peptide. Those are things that are worth protecting. The protocols you're using to purify and the data for yield improvement, your quality quality control procedures and the analytical testing methods that you're using to provide the quality product that you are, the data that you're using to scale up from the bench to commercial product. Those things are aspects of manufacturing know-how that you would not want your competitors to possibly know. And lastly, as another example, is your stability data, your storage cycle details, or your lyophilization, things that are helping with your actual peptide that you would not necessarily want people to be able to practice very easily. And so how do you make the determination between what do we patent and what do we trade, what do we file as a trade secret? So this chart here lays out some scenarios that might be helpful as you're thinking about what do we keep as a trade secret or what do we patent. So you may choose to patent a particular process if you can get claims that cover the competitor's process. Again, a patent is the right to exclude others from doing things. And so if you have a novel process, but you know that a competitor is also doing a similar or even the same process, once you have claims over that process, then you're able to get protection against that competitor using your process. We also talked about licensing revenue. If that is a goal to either cross-license or fully license your process, then having a patent can be a valuable tool to allow for that. You might also want to patent the process if a competitor can independently discover the same process. With the patent, if there is independent research or even reverse engineering, you still have a cause of action against a company or individual that is doing your process. That is not going to happen if you have only kept it as a trade secret. There might be some civil actions that you can file against either an individual that took that formula or came up with it. But in terms of an action specifically on the trade secret, unless you know that someone took your trade secret and then went to practice it, if they individually came up with the idea or they even reversed engineer it from your product, then your value as a trade secret is potentially gone. What is the risk of filing a patent? Well, it could be challenged and invalidated, and you've also fully disclosed. Now, there's another option with regard to when would you want to keep something totally as a trade secret? And that is when you can't reverse engineer the final product. It is a very proprietary, very complex way of doing things. And you understand that even if they have your product, they're not going to be able to reverse engineer it, or they're not going to be capable of independently coming up with that process. Then that is something you may want to keep as a trade secret. Or if you think your disclosure is going to give your competitor a roadmap towards your product, you might want to keep the process a trade secret. Or if you think that advantage might last beyond 20 years, we know that peptides and medicine technology is evolving daily, but your process might actually be something that goes beyond the individual product, and you might want to consider keeping that as a trade secret. But as I touched on before, there are risks. So a reverse engineering of that process, your trade secret loses some value. If there's an inadvertent disclosure, not one that's purposely done, but it just happens to be disclosed, then again, it reduces your trade secret. And then lastly, if there's employee departure, that adds to the risk of maybe that inadvertent disclosure getting out to the public. And so how can you take advantage of the patenting system as well as using trade secrets? And one particular strategy is the layering strategy. So you get the best of both worlds. You can patent the broad process architecture to establish enforceable rights and create licensing leverage, while also maintaining the specific optimization parameters, the details that make the process work at scale. And you can keep those as confidential trade secrets. It is a common practice for peptide manufacturers to kind of balance getting that public exclusivity with the confidential competitive advantage. But there's one thing that you should be aware of, and it's a key warning, is it is possible if you use this strategy that in your filing of the patent, you over disclose. Patent law does not require you to spell out the exact formula for what you're doing. You have some requirements that you disclose what the invention is and how to make it and use it, but you can use language that's a little bit more general and doesn't require you to tell the exact specifics of what you're doing, such as the type of vendors or type of suppliers that you're using, the specific chemical, if a group of chemicals would do the same job, you can disclose it that way. And so if you're in a situation where if someone can do your invention or do your process just by reading your patent, you're more than likely over disclosed. And so talking to counsel and having a strategy on how best to describe your invention without divulging your valuable trade secrets gives you those two layers of protection that are valuable. And so this chart here just gives some examples of what you might want to patent versus what you keep as a trade secret. So the novel peptide sequence you might want to describe in your patent, but your fermentation or cell culture conditions, those are things that you might want to keep as a trade secret. So as an example, the exact pH and temperature, things of that nature. You might want to patent the method of synthesizing the peptide, but the way that you purify it, the specifics of that purification process, you keep as a trade secret. For a claimed therapeutic use or mechanism of action, you might want to put that in the patent, but the specific formulation recipe and the specific ratios that give you your product, you might want to keep as a trade secret. So you might put in a range of ratios or categories of compounds that work with the particular formulation without giving your exact commercial product. And lastly, if there are any structural analogs or variants with documented improved stability or bioavailability, you want to try to protect that with your patent while keeping as a trade secret those supplier relationships and raw material sourcing specifications that are invaluable for your business. And so this layering strategy can provide you with two aspects of protection of your intellectual property. The third pillar, which I'll go through pretty quickly, are trademarks. And so trademark is a name, logo, symbol, or combination that identifies your company or product and distinguishes it from your competitors. It could be overlooked as the third pillar. It's number 3, not by lack of importance, but that's just the way we ordered the presentation. But it's very important and should be thought of with your whole IP strategy because, like trade secrets, these trademarks do not expire. As long as you continue to use it and you maintain that registration, that trademark will live much longer than the term of your patent. And so as an example, drug brand names are trademarked because they're distinct from the generic and scientific name. So when you're choosing a trademark, you want something that has some distinction. A generic name or even using the chemical name may not even receive a trademark because if it's generally known and it's not associated with your particular product, it's harder to get a trademark. But if you have a distinctive name that if one looks at it, they would know that it's your product, that can provide some serious value. So company names, platform technology, those are protectable. Trade dress, I'll use my Coca-Cola analogy again. The bottle, the Coca-Cola bottle, that is something that Coca-Cola has protected because when you see the Coca-Cola bottle, you know that is produced by that company. And so as companies are thinking about protecting their peptides and how they're marketing and packaging, if there's something distinctive with the actual packaging, you may want to consider filing a trademark to protect that. And so the critical message is that your patent will expire after 20 years. There are strategies to try and lengthen that protection, but ultimately, once you file a patent, 20 years from that filing, you're going to lose that limited monopoly that you receive from a patent. But your brand is going to carry on well beyond the filing of that patent or the expiration of that patent. And so while the introduction of competitive products can provide some strategy decisions on how you continue to market, if you've built a strong brand and your trademark is still maintained, you can still keep that brand going because you've built genuine customer loyalty and people recognize that brand. And so here is a quick reference of the three pillars that we talked about. Not going to go into each cell here to talk about what each does, but a strong takeaway is that when you file a patent, you get protection for 20 years from the filing date. A trade secret is indefinite as long as you keep it secret. And then a trademark is indefinite as long as you use it and maintain it. And at the bottom there, we give some suggestions on why you should use each of these particular pillars to protect your intellectual property. And so innovation that can't be easily designed around. That would be for a patent, things that can't be reverse engineers. That would be something that you might want to keep as a trade secret. And then as you're building your brand, you want to make sure that you have your trademarks protected. And so new science, new IP challenges in two areas we're going to talk about. I know that there are other areas, but we're crunched for time, so we're going to focus on these. The areas of personalized medicine as well as the use of AI, particularly generative AI. And so two of the most exciting frontiers in peptide therapeutics are personalized medicine and AI-driven peptide design. But with these frontiers, there are complex IP challenges. Patent law was built for a world of standardized products, and as technology evolves, the law, while it's evolving, it's not evolving at the same speed as the technology. And so sometimes, or the law and the decisions that affect the law have not necessarily caught up with that aspect of individualized medicine or using AI to help generate new candidates for peptides. So starting first with personalized medicine, when a treatment is specifically designed for a single patient, the patent system creates a significant obstacle. As we laid out in the discussion about patents, courts have ruled simply that applying a natural biological relationship to a patient, for example, if the patient has biomarker X, given peptide Y, is too close to patenting a law of nature, is not allowable. And it's particularly challenging with peptides, as some of those sequences are similar to those that occur naturally. And so courts, as well as the patent office, have turned a sharp eye on those type of claims. There's a lot of jurisprudence that came out after decisions like Mayo and Alice, and there are guidelines that the patent office uses to determine whether or not you are trying to patent something that is either naturally occurring or an abstract idea, and there are elements that you need to show there. And so while patenting the specific peptide that was created based off the individual patient has some challenges, you may be able to patent it, but it might take a while or it might be very difficult to do. There are other aspects to consider when you want to file a patent on something that was created using personalized medicine. And so the method or the process that was used to identify which peptide to make for a specific patient, that's something you might want to file a patent on. The manufacturing process for producing the personalized peptide, any novel structural features of the peptide that can be applied a bit more broadly. If, for example, a novel chemical modification that improves stability while it was developed for an individual, through research, you understand that this could actually be used a bit more broadly with other patients, or it might lead to other developments. And that's something you might want to focus on, as well as a diagnostic test to identify which patients are candidates for the treatment. There might be a little more that you need to claim beyond just the test of identifying candidates, but it is an area working with an IP counsel that you may want to focus on, especially if you feel like this particular test is novel and not obvious to those out in the world. Now, switching over to AI as a tool. AI designed peptides, it's very important to remember that the human needs to still be a dominating factor in deciding what the invention is, or coming up with what we call conception, or coming with the formulation of what the peptide is going to be. AI, from the patent office, their definition is that AI is a tool. An inventor can only be a human. And so as AI tools are continually being used in research, we're being asked, even in a law firm setting, to use AI to help us become more efficient and figure out new ways and to think critically about some of the problems that our clients are addressing. We know that you're also using these tools to come up with the latest innovations and the latest medicines and the greatest candidates for peptides for treatment of diseases. And so as use of AI tools continues, the patent office is going to be focusing in on where does the human element of this development begin? And is that the critical piece, or did AI actually come up with the full concept? There have been a number of cases where it's decided AI cannot be an inventor. Human is a human being has to be named. In November 28th of last year, they've come out with new guidance with regard to how the Patent Office is going to interpret AI inventions or inventions that utilize AI. And the precedent is that the human elements and human contributions have to be at the forefront. And so for a practical standpoint, the scientists that have directed the AI, that chose the target, that set the parameters, that interpreted the results, these are things that need to be documented and shown that you're not using AI to actually invent the peptide, but rather is using as a tool to make you more efficient in your own research on which peptides or which drugs are valuable for a particular treatment. And so there are going to be questions and challenges with filing claims to a particular peptide, asking whether or not the human came up with that concept or if it was generated by AI. Then you want to think about the AI model itself. There is value in the data that you're using, the model architecture, and the parameters that you're using. It may even be more valuable than the individual peptide that you generate. And so how do you protect that in addition to the peptide or the process that you've developed with the use of AI or the help of AI, I should say. And so we're back to layering, right? We're back to you can file certain things as a patent, you can file certain things as a trade secret. If it's in, depending on certain parameters, it might be valuable for you to file both. So you might want to file a patent on the platform process or the manufacturing methods or any of the novel peptide structures that are broadly applicable. When it comes to the AI, the specifics of the AI model, you might want to keep as a trade secret so that a competitor can't easily replicate those disclosures. And then again, the layering strategy, if there's broad process architecture, you can file a patent on that, especially if you're documenting how the human is contributing to that, but also keep certain things as a trade secret. And so again, here is an example. I'm not going to spend too much time on this, but again, as the problem with that is presented by personalized medicine is that it might be considered naturally or there might be a natural biological relationship. And so there are going to be challenges with filing a patent on that. But there are aspects around even that individual peptide that you could file a patent on, such as the platform or the process that generates the treatment. Similarly, AI tools, they're going to be questioned. The patent office or courts are going to want to know where the human has invented this particular peptide or process, so that it's not a situation where AI actually did the inventing. And so you want to document. Scientists are already in the habit of using lab notebooks to document their innovations and to track results and things of that nature. And so you want to continue to do that. And then also think about if you've developed your own AI model, keeping that a trade secret and protecting it that way. And so future-proofing your portfolio. Peptide therapeutics are evolving rapidly. Your IP strategy also needs to evolve as well. Yes, you want to protect your lead compound, but it might not be sufficient to protect your business tomorrow and the days in the future. And so you want to stay ahead by watching the technology horizon for new patentable opportunities, while also managing the full life cycle of your existing IP portfolio. And so IP lifecycle management, or what we call the patent cliff, is important. Magda touched on some of these ways to protect, especially with your composition patent. So you file your primary patent on the composition, then there are secondary patents on the formulation, the process, the dosing, the combination. Don't forget about regulatory data exclusivity. That is something to keep in mind. Here we are to pillar 3 with regard to your trademark, how you're marketing and how you're protecting that brand and building that brand so that you have value even beyond the life cycle of the patent. And then you're regularly doing audits to see where there are potential gaps, where protection might expire, and where there are new opportunities to file to continue to get protection. And so you need to plan your IP strategy at the launch for the full product cycle and not just the first patent. So having some real thought about how you want to protect this holistically, as well as the companies that manage the patent cliff deliberately, they maintain far stronger market positions than those that address it retroactively. And so thinking about these things as you're going into your development is important, because if you miss a step, you might miss an opportunity to protect something that can provide value to the company. And so key takeaways, and I'll turn it over to Magda to provide a few comments on takeaways, and then you'll hear me again.
Yeah, just really quickly, again, a layering strategy. Rashad, do you mind going back to, the layering strategy, patent, trade secrets, and trademarks, and provides the most durable competitive protection for peptide companies. Patent scope, we discussed, is a strategic decision. Broad claims require broad data to support them, and well-structured portfolio has protection at multiple levels of specificity. And then filing timing is critical. Establish pre-publication review process. Keep track of everything you're doing and file before any external disclosure, particularly if international markets matter to your business.
Yep. And as I stated, personalized medicine and AI-driven discovery, they require a shift in strategy. And so you may want to patent the platform and the process, not just the product. And also think about trade secrets when it comes to your AI models that you're developing internally. Manufacturing know-how is a core competitive asset. And so deliberately choosing what aspects of that manufacturing know-how to patent versus the protection you get from a trade secret is very important. And you may want to consider layering those as we laid out before. And then lastly, your IP strategy has to evolve with the technology. We suggest you conduct regular portfolio audits. You actively manage your patent lifestyle cycle to address the patent cliff before it arrives. You want to be proactive rather than reactive when it comes to your IP strategy. And so here are 3 things that you can do this week. I know the week is about over, but maybe it starts next week, so maybe on Monday. So identify one process or innovation in your company with no formal IP protection and determine whether it should be patented or formalized as a trade secret. Check whether your team has a pre-publication review process in place. If not, how will that look for you? And maybe we can assist you with that, or talking to your own IP counsel about how to put that together. And then lastly, reach out to your IP counsel and see when your next portfolio audit is scheduled. If it's not scheduled, get something on the schedule. And so we thank you for your time. And now we will turn it over to Jonathan. Thank you very much.
Thank you, everybody. There's one question that came up just during the end of the session, so maybe we can address that one before moving to the next step, and then I'll have a few questions towards the end, if you don't mind. It's Derek who asked, how do you structure your claims for peptide sequences to cover as many analogs as possible?
So my suggestion, and I'll also let Magda provide her comments as well, is most importantly, your claiming strategy, especially with regard to peptides and analogs, is going to go right along with the data that you have to support it. And so while in theory, you might be a number of different analogs might work for the particular purpose that you're using it for, courts have now honed in on the breadth of those particular claims. And so while you want to cast a fence as broad as you can, if you don't have the data or you can't generate the data, one important feature of US patent law is that you don't have to have the data right away. You can provide some supplemental information with regard to that data. But if that data can't be generated or will not be generated, you might not want to try and claim your peptide sequence as broadly as you think. The data is going to help guide that way. That's not to say you can't take a risk and potentially draft your claims a bit broader with hope that the data will be there. But you do want to be careful of those considerations, because if you try to claim a universe where you can't support it with data, it's potential that your patent could be invalidated.
And there's some language specific language in the claims that could be used to, you have your broader sequence, and then you can say, and derivatives or analogs. But like Rashad said, I mean, there has to be enough examples, enough data to support that kind of claim. Patent office sometimes when they see or a derivative or analog of, they might not like it. But if you can make an argument that you have the data, you have support, you provided enough examples of these analogs, there might be some, functional features too that, you can claim the analogs through, their function. So there's different ways, one can go about this, and, it's, again, different layers, different strategies. But yeah, so you have to be careful, but there is ways that you can accomplish claiming analogs or derivatives.
Thank you. Yeah, and that was there was a follow-up question from Derek that came up, but that was exactly what you just said. So, thank you. And I also had a question while we're at it, and then maybe I'll pass the word to Jonathan. But it was, since you mentioned that in the US specifically, the system is first to file. Is it different in different countries? And if so, is there a consideration to be had? Like, do I file in the US first, or do I file elsewhere first, or is that even a thing?
So the US actually used to be first to invent. And so we've actually changed our system right around 2013 with the American Invents Act so that we are now in line with the rest of the world. And so it's important for us to remind people that we've gone from a first to invent system to first to file. I know it's been almost 13 years, but patent law has been around much longer than that. So the first to file system requires you to answer some questions and to get to filing your patent a little sooner than maybe first to invent, because you could have proved that you invented something before somebody else. But you also need to use that caution in making sure that your disclosure will meet those requirements of the patent law. And so as you're considering your global strategy, because we're now more in line with the rest of the world, your strategy in terms of filing in other countries won't change that much. But there are some other factors you may want to consider, such as where your main inventors are. For example, if they're based in Europe, you may want to file first in Europe to get a filing, and then that filing could be used to support your filings around the world.
Okay, thank you. I haven't seen any other questions pop up in the Q&A just yet. So maybe we can. Oh, sorry. There's just one that just came in. Priority date was mentioned in the context of initial documentation of an invention. What exactly does priority date mean?
So a priority date is the date where you have your first filing of a patent application. Why that is important is that once that date is established, any reference or prior art, as we use in patent law, cannot be used against your claims in your invention. So it's important to get an early priority date so that you narrow in the amount of references that might be applied against your patent filing. The earlier you get your date, there's less references that could be created. But again, you want to balance that need to file early with also having the proper disclosure to make sure you get the claims that you want. And so a proper strategy is definitely focusing in on getting the earliest priority date that you can, but also focusing your patent drafting strategy so that you can support any disclosures that you put out there. But you definitely want to cut off the amount of references or prior art that can be used to say that your invention would have been obvious, or it's not even an invention at all. It's not novel at all.
So priority date is just when you file.
Correct.
Thanks.
That's great. Thank you very much. I don't see any other questions just yet, so maybe we can move on to the next bit. And if you have questions regarding any part of the presentation, we can always cover them after Jonathan's talk as well.
Oh,
So,
Alec, I
Jonathan.
just want to ask. I just want to ask sort of a follow-up question to the first to file question. So, in the U.S., it moved sort of midway, but mostly aligned with the rest of the world to first inventor to file. So, my question for the attorneys is: have you seen elsewhere in the world where it's just a pure first to file, and others can file without being the inventor?
I think you guys are
Sorry.
muted, Rashad or Magda.
Or Rashad, or Magda, do you want to take a stab at that?
So I'm not sure exactly like if I understand the question correctly, but uh, first to file, that's sets your priority date. Most of the world follows that rule. Um, and I mean, there has not been, other than the US, the rest of the world, follows that kind of strategy. That's your, priority date, the date that counts. Like Rashad and mentioned, that's important for, counting the prior art, and making sure that, you avoid any of that. And then, what was the second portion of your question?
Oh, that was the, I guess I was curious about and maybe this is more um semantics rather than actual substance is so the US moved to a first inventor to file. So it's kind of I guess it used to be first to invent, right? And now it's first to file largely, but still first inventor to file in the sort of the letter of the law. I was just wondering if you know of countries in the world where it's a pure first to file and others who may or may not have had the invention could file a patent. Like I mean this is probably more theoretical than anything else.
Shad, I'm not sure.
That's fine. We can move on. This is
Yeah,
yeah.
there are countries where, a lot of that's going to depend on your business's priorities as well. Um, Having an understanding of how each country takes a disclosure is important. For example, I worked with a client that they filed first in the US, and then realized 16 months after filing that they wanted to file in Australia. Well, the reason why that's important is that in the US, your application is going to publish 18 months after you file it. And
Right.
so they were able to file in Australia because Australia's grace period allows for you to file before there is a public disclosure. And the only public disclosure would have been the publication of the US patent application. And so while all countries at this point, I'm not aware of a country that does not have first to file, where your strategy might differ or an understanding in terms of, yes, I disclosed here, but can I still get rights, is the understanding of those particular grace periods. You would not have been able to file in Europe if that's the case. They have what is called absolute novelty. And so once your first disclosure, whether it's the filing of a patent application or speaking at a trade or association or a conference, you need to have something on file. But like I said in my other example, if Australia is the key and you're not going to have anything published, you could still get something filed with that grace period.
Would you then recommend like a international pen in that case? Like would you recommend that over like filing individually in every country?
I think a good piece of your strategy.
Yeah.
You definitely want to make sure that you get that first filing, like Magda talked about with the provisional. The benefit of getting that priority date is that with the patent cooperation treaty, they're going to recognize that first filing everywhere. And
Right.
so if you know that you're going to look into maybe commercializing or manufacturing in a number of different countries, then going the patent cooperation treaty route is the best route, I think, because it gives you the opportunity to enter so many different countries while also just filing that initial one patent application. However, if your business is limited solely to the United States or to Canada or maybe just a few other places that you're looking at, then it might be more ideal to file directly in those countries. From a cost standpoint, it might end up being cheaper than going the PCT route, but also it might fit more into your business strategy. So if you're looking for global commercialization, looking at an international application is probably something you should consider very strongly. But if there's only a few markets that you're looking at, and you don't need to have the option of filing in over 150 different countries, then filing directly should be a part of your strategy.
Yeah, that's
Just
very sage advice. Yeah. But I think most usually pharma when, and this is sort of, putting the investor hat on, usually pharma when they look to acquire an asset, they're looking for international rights across many different markets. So something to think about when you're filing your patent as well. Yeah, but very sage advice and great presentation. It really was a masterclass in the legal aspects of intellectual property. Yeah. So with that, I'd like to if um, invite Jonathan to come deliver his piece.
Thanks very much, Sally. Okay, so I'll just share my slides. Okay, so what I'm going to talk to you about is the scientist's role and the scientist's viewpoint of IP generation. So the previous talk was absolutely excellent in showing all the advantages, whereas when we're generating data, there's obviously a role for the scientists to play and a number of questions for them to be asking themselves. And this is a general walkthrough of the typical things that we would ask ourselves, and we'll run through some opportunities for generation of key and valuable IP outside of the substance and matter of the compound. So essentially, the scientist's role in IP generation is fairly simply summarized in one sentence. They must generate and summarize the key information to generate a strong patent. Now, that's very easy to summarize, but what actually constitutes to that invention and strong patent? And we need to recognize, is the product or formulation new? Is it novel? Does it have novelty? Does it contain a non-obvious or an inventive step? And are there clear advantageous properties or surprising results with the invention? We'll run through some of those examples. Enabling, is there enough guidance and examples so that in practice the claimed invention can be repeated or re-prepared without significant experimentation, by someone skilled in the art? And from a written description point of view, you want to make sure that there's a representative number of species to prove the full possession of the scope of the claimed invention. And I think some of the previous discussion regarding that is excellent. We need to make sure that our claim is broad enough, but not too broad. We need to make sure that it's specific, and we need to make sure that it's well documented. So, what scientists can do to bolster this inventiveness is essentially ask themselves a series of questions. Starting with, what is it that we're trying to solve? Outside of the composition of matter, what challenges have we approached? And do those challenges come from things like the yield or the purity, is poor, and we start to introduce an inventive step there? Is there a particle size challenge? Do things dissolve too quickly or too slowly? The crystal habit, are they impossible to then formulate, or are they too easy to formulate, or do they settle? Is there a challenge with handling, which then feeds into bulk density? Do we have a challenge with solubility or dissolution? And is there a way that we can approach that? And we'll come on to that. Is there a challenge with regards to stability, which is a key challenge that we come across routinely when we're working with peptides? That the stability of a peptide in solution can be quite challenging, or in some cases it can be excellent. Is there a trick, or is there a change to the molecule that can support us there? So once we're looking at these problems and we've defined our problem, then we start to consider what experiments or formulations are there available, and are there experiments or formulations that worked and those that didn't? And then if there are ones that worked and didn't, what's the difference in properties? And then what was the ultimate solution, to solving that challenge? Did we look at combination with other therapeutics or other compounds, as in salts, for example? is what we're solving leading to an improved patient experience, or is it leading to things like enhanced stability, solubility, PK profile, etc.? And then we need to compare how we've solved that problem to what is available out there in prior art. And I'm going to run through just a couple of opportunities, but these opportunities then would fall outside of the substance of matter. They would fall within this secondary product pillar, of which we can see crystalline form, so salts or co-crystals, enantiomers, metabolites, and formulations. And essentially what we're looking to do here is we're looking for patent life extension, and we're looking to support and bolster our IP position. So if I just run through the first example of, peptide crystallization. So I'm fairly sure everyone would understand what I'm referring to when I'm referring to a crystalline peptide. But what we are referring to is a repeating regular unit of peptide subunits within the solid state. And we're relying on growth of those in a periodic arrangement to avoid a random arrangement of peptide, which then leads to something like the gel on the right-hand side. So if we are taking advantage of intermolecular and intramolecular interactions, such as, pi-pi stacking, hydrophobic interactions, or van der Waals interactions, and hydrogen bonding, then we can lead ourselves to what you can see on the right-hand side of the slide there, where we have a crystalline product, and that crystalline product itself has advantages. Those advantages of an isolated solid form outside of lyophilization or spray drying are that we can overcome some pretty critical challenges with regards to peptide synthesis. So, we can overcome challenges with regard to column purification. Column purification can be inefficient. It can be also very efficient, and it can be very, very good at rejecting certain impurities. Crystallization can give us the added advantage of isolating as a solid rather than isolating by it through a column and then isolating by lyophilization or spray drying. We can also give ourselves enhanced thermal stability, and that may allow us to then have room temperature storage, for example, instead of cold chain. We can have decreased hygroscopicity, and again, that may allow us to remove a desiccant from the packing arrangements. Or we may have a consistent mass balance where we don't have to consider the amount of water and run a KF before we do an experiment. It can lead to improved particle handling where things are less fluffy and improved density. Or we may encounter things like modified or decreased dissolution rate, where we may have a stability enhancement. And it's this modified or decreased dissolution rate where there's some considerable interest in peptide crystallization when we're referring to oral peptide deliveries. So the typical way of doing this is, from a top level, is doing a series of small scale crystallizations. We then step up from plates where we're doing small crystallizations to a 1 to 2 mil screen. And then we start to look at some scale up crystallizations. And essentially the way that we do that is we generate the crystalline peptide on a small scale, and we define those nucleation conditions. Those nucleation conditions themselves are non-obvious because we have to refine ourselves. And crystallization of a peptide is a tricky business. So, it needs to be done by someone who is skilled, and you don't essentially stumble across the conditions for peptide crystallization. So there is a considerable amount of knowledge and experience that goes into this. And then once we've found those nucleation conditions on a small scale, we then go to 1 to 2 mils, and then we scale up to a jacketed reactor. But the way that we do this is, you would take a crystallized peptide as a seed. So, we've generated something on a very small scale, and then we would suspend it on a fiber or a horsehair. We would then vortex those crystals and then dilute and then use in multiple new experiments. So we're going from a single well, which may contain microgram of compound, all the way through into vials where we can then have milligram, and then into reactors where we can have gram quantity. And that progression of isolating and first off crystallizing requires a series of solid decisions on solvent composition, acid or base or buffer, as well as things like polymers or templating agents, etc. So, there's real critical information that goes into this. And you can see just on the schematic here that we can go from a lab scale all the way through to a manufacturing scale. And there's plenty of examples of patented crystalline peptides, and as you can see just on the slide here, from 2025, 2019, we've got a good number of examples where within those patents, the disclosure is the method of crystallization as well as the advantages of the solid form and key differentiating features of that crystalline peptide, including ways on how to make it and how to store it and how to use it, essentially. So to just run through a top level example of, some of the decisions that are made when we're looking into crystallizing a peptide, to give us that obvious advantage for a solid form is, many crystalline peptides are cyclic. They're cyclic because they have a rigid core. So I would advise that if you do have a cyclic peptide, it's something to consider and do some experimentation in your lab. But the reality of it is that when you're crystallizing a peptide, you don't end up with one crystalline form, and you don't end up with a single non-solvated, non-hydrated crystalline form. You can see on the table here that you actually generally get something which is solvated or hydrated. This example is a peptide of between 1200 to 1400 molecular weight and isolated as an acetate salt. But you can see that the peptide could be isolated, solvated, an acetone solvate, an MTBE solvate, cyclohexane solvate, isostructural solvates containing things like DMSO, ethyl acetate. Those are common process solvents, or we can have unstable, metastable forms. We can have solvates where things have changed. So where we've isolated one form and then over a period of time, the solvent of crystallization is then lost and we've generated a new form. And you can see that from a process point of view, including any of these solvates within our pattern then shows that, we have specific control on a crystallization process. And if there is an obvious advantage to isolating, say, for example, a cyclohexane solvate, because it rejects a specific impurity or gives us a good flowable product, we know that we can remove that cyclohexane. We know that through further processing, we can then go back to the native peptide without any solvent of crystallization, which is a key advantage. And this is typically the way that we would look at the inventive step of generating a crystalline form. So you can see here that on the schematic on the right hand side, typically the way that a peptide crystallizes, you will end up with either discrete areas of crystalline peptide within the unit cell, and they may be regular repeating with solvent in between, as on the left-hand side, or there may be a channel type structure on the right-hand side. And I would say in the general sense, the channel type structure is more common, where that solvent of crystallization actually sits in a very discrete layer within the solid form. And that has the advantage of, we've crystallized our peptide, we have a solid form, but then that solid form, we would then need to then dry. But the drying is actually pretty straightforward, using normal processing equipment. And it can be done, either using dry or ambient vacuum. Or it can be then done by the introduction of humidity or water. And, in this example, where we've got a number of inventive steps for the crystallization of this peptide, where we started off with the amorphous form. That amorphous form was actually generated by lyophilization during the process. And then through some experimentation, you can actually see that by slurring in ethanol, we then end up with type E. Type E itself is an ethanol solvate. That ethanol solvate can then dry to type B. And, by going from the amorphous form where there is no regular repeating unit and there is nothing novel about that solid form, we can then introduce a key inventive step to getting ourselves to a dry crystalline form that does not contain solvent of crystallization. But the key advantage actually to this example is that we could actually improve the purity of the isolated material through generation of this solvate. So through generation of the solvate, you can see in the table on the bottom of the slide, you can go from a 90% or lower purity all the way through to 97 or 96% by isolation, and then there is an increased stability in that product. So if we go from an impure product, we reject an impurity, generate a solvate, and then dry that, we have enhanced stability. And you can also look at things like rejection of a specific isomer or specific impurity from these types of experiments, where, for example, one experiment we had a beta isomer where we went from 14% down to 6.5%, and then through refinement of the process and the crystallization process, we can actually then go from an even lower percentage, so 1.5% down to 1.2. So if you're thinking about building the story for your in-house patent attorney or for your patent counsel, there is an obvious progression of, we isolated the material from ethanol, we generated a specific ethanol solvate that rejected a beta isomer, and then we can dry the product and then remove that crystallization solvent. And you can see here that across multiple experiments, so starting on the left-hand side of the graph, you can see that we had a variety of different batches, each containing high ethanol content, upwards of 26,000 ppm. And then by drying under humid drying, you can actually then start exchanging out that ethanol content and then work our way down to an ethanol content below ICH guidance. And actually here, using 33% relative humidity, we could actually reject that water. And then that does have the added advantage of actually further drying under 33% relative humidity, then pushes the material to then return to its amorphous state. So, not only have we isolated a crystalline form, which is non-obvious and inventive, we've then generated a solvate. We've then dried that solvate into a new form, so form B, which then has all the advantages of then being able further handleable with a low ethanol content. We can actually return ourselves to the amorphous form by further drying with 33% relative humidity, where we then actually return ourselves back to a higher aqueous solubility and retaining high purity. So, there's a number of advantages which were unanticipated, where you can see through a full story, you then have a patentable product. So another opportunity is through peptide formulation, and there's a number of advantages obviously to peptide formulation, and you can see here where we would start with the composition of matter patent, and then we would consider ourselves, well, we may be working with a consistent or a similar structure of peptides within our portfolio. Is there a formulation platform that enables solubility or stability for the peptide, and then can we apply that to a platform? And the key advantage there is actually, the formulation platform itself is patentable, even though it's not exclusive specific product. That formulation platform then can actually be itself a patentable product, and then they can undergo improvements, and then we can apply that specific formulation to our new chemical entity, and then further expand our patent window. This is becoming quite a hot topic for oral peptide delivery routes, where parenteral delivery routes obviously rule, and you can see here the general trends from 2010 to 2018. But one of the key areas is the oral peptide delivery is growing, and particularly with regards to the GLP-1s. There are multiple different avenues, particularly with regards to improving peptide permeability, which remains an ongoing challenge. So, we may overcome the stability challenge of a peptide, but then understanding peptide permeability and improving that is a key area for advantages. And it all starts with understanding the peptide molecule. So, if you're looking at the peptide molecule, we need to understand things like isoelectric point, to then understand, is there going to be a solubility challenge? Is there an assessment of crystallinity or confirmation of amorphous nature, which we've run through in opportunity one? What is the solid form stability? Is there any instability with regards to freeze-thaw cycling? If we are isolating by lyophilization, and are there any challenges with regard to solid or solution stability to then highlight any key stability concerns? And a common challenge with regards to peptides are obviously self-association mechanisms, where we may then dose a peptide at a specific concentration, let's say 1 mg per mil, for example. But over a period of time, do we start to see risk of self-association or glass adhesion? And if we do start to see that, is our active dose actually decreasing over time? But once we have all this knowledge and all this information, we actually know what the challenge then is to be solved. So then we can approach it from a methodical, logical manner. And approaching a formulation from a methodical and logical manner generally starts with looking at things like solubility. First off, considering the mode of administration, but if we're looking at the solubility, a general list of what parenteral administration or subcutaneous may involve are things like phosphate buffers, citrates, succinate buffer, tris, carbonate buffers, saline, water for injection, or D5W. And an example of the risk associated with looking at that baseline unformulated approach for a peptide, you can see on the right hand side there, where if you took 3 compositions, so pure water, peptide in water, and then peptide in saline, you can actually see over the progression over 2 weeks, you start to actually get peptide precipitating out of solution and adhering to glass in pure saline. So we know what the problem is there. We know that saline can't be a platform formulation for this peptide. So we needed to then understand what types of additives and excipients could be added. And that then would form part of the excipient selection strategy. So if we are overcoming selective instability or dehydration mechanisms, we can look at turning to sugars. If we're having challenges with solubility, we can look at polyols or surfactants. We can even add things like sacrificial antioxidants or amino acids, metal chelators, polymers for then making sure that we are avoiding aggregation and precipitation, and cyclodextrin and lipids. So you can see that once we understand the problem, the scientist's role would be then to approach it in a methodical, sensible manner. And generally the approach would be that once we've identified the problem and we've identified an excipient strategy, then we introduce things like short-term stability to then confirm that there aren't any red flags with regards to drug excipient compatibility before we then start moving forward into more formal stability studies and formulation studies. And I'll just wrap up by giving you an example of a peptide formulation platform, and this is actually Elgard. So Elgard is actually a therapeutic for an anti-cancer peptide owned by Tolmar, and it gives a long release subcutaneous dose. And what Tolmar actually did was they actually found that during development of other peptides, there was actually the opportunity to take their formulation for Elgard and apply that to octreotide. And octreotide actually had some significant stability concerns, even though the biological activity was excellent. There were some stability concerns, and then that led itself to a short half-life. So by screening a number of different excipients and solvents and technologies, they actually ended up, turning to the same formulation from Elgard. And that actually relies on a BEPO technology, which actually then focuses on copolymers. And you can see just on the graph on the bottom there that you can see over a period of time, the acylated peptide doesn't release anywhere near as well as the native peptide. Therefore, some challenges were faced. But the platform that was turned to is this BEPO technology. So it's actually a a fairly simple technology with regards to copolymers, a well understood technology where the ratio of PEG to PLA was actually modified to then give a very specific ratio. And then very simply, the technology is that you dissolve the peptide in a common solvent, and then the copolymer is then added for controlled release, either using a diblock or a triblock copolymer. And you can see on the graphs there that the stability was significantly improved by controlling that rate of acetylation. And starting at the top, that was one example, where the rate of acetylation was very high over a series of hours, but then adding in specific metals, they could actually then change that, or by adding in different excipients like permoic acid or sodium docusate, you can actually see that they can vastly decrease that risk of acylation, which is very important to improving the stability. So, in just this one example, there was a problem that was faced with one specific peptide for Elgard, but then taking that peptide platform of the diblock and the triblock copolymers, it can then be expanded out to other products, which is obviously a key advantage. So to just summarize, the scientist's role in IP generation is obviously essential to support novelty and then introduce the non-obvious or inventive steps, which then leads to enablement and then making sure that everything is written up correctly and thoroughly to provide a full written description. And then I gave you two examples amongst a sea of examples where IP generation can be applied for peptide therapeutics. To then generate opportunities for product protection beyond the composition of matter, as in peptide crystallization and early formulations or platform formulations, which are particularly useful when peptides have common features. But essentially everything boils down to the early statement that I made, where generation of strong data is absolutely essential and key for protection of product. So with that, I'll thank you for your attention, and then I'll be happy to take any questions.
Thank you very much, Jonathan. Are there any questions from the participants? Please use the Q&A window. There is one which is for the previous conversation, so I'm just holding on to it a little bit because to see if there are ones specific to this one.
Sure, I have a question. Thank you for a really nice presentation, Jonathan. I was just curious regarding crystallization of peptides. Are there any particular features or certain size of peptide that makes it more amenable to crystallization?
Yeah, that's a great question. I would say cyclic peptides are generally easier and more straightforward to crystallize because they have that rigid core. But there are plenty of examples of crystallized peptides out there where they're oligomeric or they're linear, where you can actually then get quite strong interactions between the core of adjacent peptides through things like hydrogen bonding. The other thing to consider as well is a lot of peptides are functionalized where they may have a hydrophilic end and a hydrophobic end, particularly where there's a long lipophilic chain. They can also crystallize quite well. We've seen examples in literature, from an industry point of view, where a long lipophilic chain can actually slow itself down because we then tune that solvent composition to then make sure that the lipophilic chain doesn't want to be pointed outwards and make something like a micelle or a vehicle. So then they start behaving themselves. But from a molecular weight point of view, I would say somewhere between 4,000 and 5,000 molecular weight is fairly top end. At that point, it does start to get quite challenging.
Thank you. Other questions?
Sorry, I was muted. There was one in the Q&A, but to me, it reads more like a question for Rashad and Magda, but let me read up. If in a patent I see a list of peptides that were actually made, but the general sequence and the claims is broader, can these paper analogs be considered prior art?
So the short answer is yes. There is a strategy of claiming or drafting your patent application broader than even what you're trying to claim. And so there are particular challenges if an analog that you are developing was generically described or described in a patent previous to your filing. But it goes right in line with what Jonathan laid out. The data and the information that indicates the novelty as well as the non-obviousness of your particular analog can help you craft a strategy to protect that particular analog and as well as differentiate from a more generalized discussion. There are strategies to getting around that particular prior art, but short answer is if somebody listed it in a document, it's out there, an examiner is going to put it on you to differentiate that. But with the support of the information like Jonathan laid out, as well as a comprehensive strategy for filing your own claims, you should be able to overcome it.
Thank you. Any other questions? You can also raise your hand in the system if you want to ask your question verbally.
Yeah, I have another question for Rashad and Magdalena. I really like that you brought up AI because, of course, AI-driven peptide design is, becoming more common. And you sort of, definitely touched on it, but just wondering if you can expand on really who owns the IP, generated by some of these AI algorithm discovery platforms and like how companies should really think about protecting both the peptide sequences and the underlying discovery models.
So I'm going to give an answer that is not going to answer your question, which is we don't know. Courts are still trying to figure that aspect out. But what I would advise is particularly around the information that you're using to train your AI model. That is something that, my thought is if you're training it, you're developing the AI model, then even the outputs are owned by the person that helped to create this particular model. Courts will figure out the parameters of that, but I would advise companies to make sure that anything you're putting into your AI model is something that you can trace back to what you potentially own. Because if it's trained on data or resources that could potentially be assigned to somebody else, then you're giving another company or another person the right to say, actually, that's mine. And so the more that you can put in that's actually data you generated or information you generated, then the stronger case you have to say that even this output is owned by my company.
That makes sense. Yeah, thank you so much. In general, are there any common mistakes that you could say you've seen startups make in drafting their patent claims that come back to like later limit their enforcement? You gave somewhat some examples, but
yeah, I think one of the key examples is trying to draft your claims or protect things a bit too broadly from a standpoint of, yes, we have this particular peptide, but we don't want anybody to come into this space. And so you run the risk of not getting a patent because you're too broad. And now you might actually create your own prior art against something now that you're honing in on a particular peptide or drug. You might have to differentiate your broader disclosure from what you're actually doing. And so that's one mistake that I've seen startups make. An opposite mistake is not thinking about your IP strategy up front. A lot of the focus is on the development of the actual product, that the IP strategy happens after the development or after the development has been going on for a while. And so you may have missed some opportunities to protect aspects of that development because now you're only focusing in on it now that you have a particular product you're going towards. And so the earlier you're able to bring your IP strategy in line with your business strategy and R&D strategy, I think they all work together versus now that we have a product or now that we have a business strategy, let's now figure out how to protect it. I think all parties to that need to be in conversation so that your IP strategy matches where your business is trying to go and ultimately supports the commercial product that you're putting out.
That's great advice. Yeah, thank you so much. I've been in the first situation you talked about where you have to try to get around your own IP. That's tough.
Yep. And as a patent attorney, it's challenging because you don't want to say anything that could be used against you in a court of law to get around that art. But you also recognize that that earlier filing is not exactly what we're doing. And we
Sure.
use this strategy a bit differently. And so now we have to navigate it. It's complex, but it can be done. But, it's something that you want to try and avoid.
Definitely. Any other questions from?
I also want to ask a question around the AI. I as I advise companies on I do a lot of investing in AI ML drug discovery, and one challenge I find with them is a lot of times because their technology is very powerful, they can come up with a lot of chemical matter really relatively easily compared to traditional wet lab processes. So they end up with the problem with having a lot of patentable material, but they may or may not want to develop all of these and may or may not have the resources to develop all of these. But what do you recommend in that case as they to achieve a balance between protection and also you're keeping the costs in mind. Oh, Rashad, you're muted.
Sorry about
Thank
that,
you.
Sally. Yes, I think the challenge with using AI tools, and even if you're not developing a new drug or new chemistries, the challenge is that you're going to receive some outputs that are even beyond what you and your team could have done in that same space and time. And so there's going to be a balance in terms of what options you want to pursue. And this goes back to my comments that I made to Yvonne, is that your IP strategy and your research strategy also needs to be aligned with your business strategy ultimately, and understanding that just because there are now thousands of potential areas for us to focus on, if you understand the market and you understand, what you're trying to, what problem your company is trying to solve, I think that's going to instruct how your R&D team may pursue it. So while the AI output is giving you thousands, if you're in line with your business's strategy as well as your IP protection strategy, then maybe you narrow that down to a few hundred that are potentially viable. You also have to be careful because AI hallucinates. While it might look like something that is worth pursuing because, oh, this could potentially work, you're now in a situation where you might be going down the line of pursuing protection on a theoretical peptide or compound that ultimately might not be something that is viable. And so I think having some discussions and some understanding of where you want your particular research to focus will then allow you to kind of narrow down those limitless options that you seem to get just based on the different prompts and different data points you're putting into your AI. And so I advise to definitely use those tools, but also have a strategy for using those tools. If you're only using it just to generate potential candidates, then more than likely you're going to expend capital and resources chasing down something that may not be in the best interest of the company.
Thank you. That's really good advice.
You're welcome.
Thanks. I was just looking at the questions, but I'm conscious of time that we overran a little bit. There are no new questions in the Q&A.
I did have just one last question to bring it back to Jonathan. Was just wondering with regard to the formulation strategies and crystallization, is there a certain timing in your kind of early development, late discovery, early development, and then moving towards manufacturing, a certain timing when it's best to pursue?
Yeah, absolutely. I would say a lot of good peptides can be ruined by a bad formulation. So if you don't consider the formulation early to then overcome, because some peptides may aggregate, You know, they may have great biological activity, but they may aggregate. That's a feature of peptides. You know, it is what it is. You know, so if you then were to then consider a very small percentage of a surfactant or another excipient to then avoid that from happening, the baseline results in the PK study may then be substantially better, and then there may be a further path towards development, as opposed to running the risk of a decreased or depressed PK profile, because of a challenge that can be solved fairly straightforward. So I would say it's worth looking at early, and then the strategy for that peptide and other peptides in the portfolio may be going towards similar targets or not. There is that added advantage of we may have a platform that is building out here, you know, of a specific, you know, cocktail of excipients which then maximize solubility and stability, and then assist with frontline PK data. So yeah, I would say early.
Sure. So maybe late discovery, early development.
Late discovery is typically where we would see some interest. I would say as people get towards phase one, generally that effort can become quite rushed. You know, and we may miss an opportunity because there is a key deadline for human formulation, for example. But you know, if there is the opportunity to do it in late discovery for PK tox profiling or safety profiling, that's a good time to be doing it.
Great. Well, thanks so much. Are there any final comments, Alaric, Sally?
I don't see any new questions. No, I just wanted to thank everyone for participating, for speaking, and also everyone who joined to listen in. Thank you very much for the panelists. It was very useful. I learned a lot of things, certainly. So I'll pass the word back to you, Sally and Yvonne.
Sally, any final comments before we close? All right. Well, thank you so much to all the speakers. Really appreciate your time and giving us all this knowledge. Definitely very useful.
Thank you.
And thank you for everyone who joined. Just a friendly reminder that we have our in-person meeting September 28th to October 1st in Napa, and hope everyone can join us. And look forward to seeing you there. Thank you again.
Thank you.
Yeah. Thank you so much,
Thanks,
everyone.
everybody.
Thank you
Have
to all
a
the
great end of the day.
speakers.
Bye.
Thank you. Bye.
Thanks, everyone. Bye
Thank you.
now.
Bye.
Thank you.
Good
Bye for
rest
now.
of your day. Bye.